Welcome to the 12-3 podcast, which is on RNA and protein synthesis, and we're going to go through several processes that involve DNA and RNA and the making of proteins. So first thing we're going to talk about is we know what genes are. We've done that before. And the DNA, the genes, can be copied by RNA from the DNA, and from there what we can do is then decode it into proteins after we get the RNA copy to the ribosome. So RNA is a long chain of nucleotides, just like DNA, and each nucleotide is made of a 5-carbon sugar, just like DNA, phosphate group, like DNA, and also a nitrogenous base. And here's a picture of DNA right here. Here's a picture of RNA, and right away you should notice a couple differences. The three main differences: the sugar in RNA is ribose, not deoxyribose. The RNA is single-stranded. See, there's only one strand here. DNA has 2 strands. And lastly, RNA contains uracil, but not thymine. So what are the 3 main types of RNA? There are messenger, ribosomal, and transfer RNA. The messenger RNA carries copies of the instructions for assembling amino acids into proteins, and it takes this from the nucleus to the ribosome. Ribosomal RNA is part of the ribosome along with proteins. Lastly, transfer RNA transfers each amino acid to the ribosome. Here's the amino acid right here. Brings it to the ribosome, where the ribosome then does its thing with the amino acid to make a protein. What's transcription? Transcription is when RNA molecules are produced by copying part of the nucleotide sequence of DNA into a complementary sequence in RNA. And this requires the enzyme RNA polymerase. If you remember, we were talking about DNA polymerase. During transcription, RNA polymerase binds the DNA, separates the DNA strands, and then uses one strand of DNA as a template to make the nucleotides that compose of RNA. RNA binds only to areas of the DNA known as promoters, and this tells the enzyme polymerase, RNA polymerase, where to bind to make RNA. Where in the DNA do you want to bind to start making your genes? So here's your RNA polymerase right here, this gray ball. It's opened up the DNA strand. Remember, these are hydrogen bonds, so they're easy to open. And what it's now doing is assembling a length of RNA from this exposed DNA strand, just this one side. And it's using uracil instead of thymine. So if you notice, there are orange bases here, and there are no orange bases here, but there are kind of this yellowish beige colored base. So in eukaryotic genes, you have these sections called introns, and they're not involved for coding for proteins. And the sequences that we use for proteins are called exons. And your genome is filled with these long pieces of DNA that really don't do much for you. So what happens is the RNA molecules copy both the introns and the exons. And then the introns are cut out of the RNA molecules, and then the exons are spliced back together to form messenger RNA. And that's what this little M stands for, is messenger. So once again, the genetic code of the mRNA is in the nitrogen bases. The code's used right with 4 letters, A, U, C, and G, just like DNA uses A, T, C, and G. So codon, this is very important. Codon is 3 consecutive nucleotides that specify a particular amino acid. Here's one codon right here, AUG. Here's the second one, AAC. Each codon specifies one particular amino acid and then that acid is placed on the polypeptide chain in the order that the codons appear. And some amino acids can be specified by more than one codon. there's a limited number of amino acids, as you can see here. I think right here you can see them all, and some of them are repeated. But you can see with 3 letters and three characters, 1, 2, 3, and a codon, and then there are 4 letters for each character. You have, I think, 64 combinations of letters, and there are only 20-something amino acids in the human body. So you can see some of them are going to code for amino acids twice. So here's how you read this chart. The first letter is the first letter in the codon, first nucleotide in the codon. The second letter is the second one, and the third letter is the third letter in the codon. So let's say, for instance, we had GCC, then that would be alanine. So there's one codon, AUG, that either acts as a start codon or methionine, and that one is let's find it, AUG. You can see it's right here. And then there are several stop codons. Here's one right there, and there's 2 more right here. And these tell us the end of a polypeptide. That's when the messenger RNA knows, hey, I'm done. This is the end of the gene, and I can stop making the mRNA. So let's look at the sequence here. We have UCG. We have 3. Well, let's see. We have this mRNA sequence. We have 3, reading 3 bases at a time, a codon. We have this. And then the codons represent the following amino acids. Let's look at this one. U, C, G. That's serine right there. C, A, C. That's histidine right here. And then GGU, GGU. That's glycine. So this chart is not all that hard to read. What's translation? Translation is when the messenger RNA is decoded into a polypeptide chain. So far we've done transcription, which is the creation of an mRNA by copying the information of DNA. Now this is translation, where that mRNA is then taken and decoded into a polypeptide chain. And this is where the ribosomes come in. So what happens here is that the cell uses information from the messenger RNA to produce proteins at the ribosome. So here's what it looks like. Here's our messenger RNA. It's leaving the nucleus through the pores. So it's transcribed in the nucleus, transcription, and then enters the cytoplasm and goes to a ribosome. The messenger RNA attaches to the ribosome, and then each codon in the mRNA, remember that's 3 bases in a row, moves through the ribosome like a machine, and the proper amino acid is brought to the ribosome by the tRNA. And in the ribosome, the amino acid is then transferred to this polypeptide chain. Let's look at a transfer RNA molecule a little bit more. It only carries 1 amino acid. Here's our amino acid right here. And it has the 3 unpaired bases at the bottom, and we call this the anticodon. These are the 3, and they're complementary to the mRNA codon. So if the mRNA codon has thymine, guanine, and cytosine, then this is uracil, guanine, and cytosine. And this one is going to be adenine, cytosine, guanine. So if you look here, here we go. Here's uracil, adenine, cytosine. Let's start with the codon. Adenine, uracil, guanine. So the anticodon that should match up to it would be uracil, adenine, cytosine. And then this polypeptide up here, this amino acid right here is then added to the chain. And you can see as the ribosome is going to move along the molecule, and as it does, it's going to start to chain the amino acids together into a polypeptide, and then the transfer RNA goes away once it's released, and then it goes and looks for another amino acid to attach to. And here's the next transfer RNA moving into place. And as this goes on, now the process goes on until we reach one of those 3 stop codons, which we talked about before. And So once again, we talked about the DNA being the master plan, and then RNA is they call it a blueprint. I call it a copy. The DNA never leaves the nucleus. It's much too important to leave the nucleus where it could be lost or damaged. So the RNA is basically a copy of just the gene. The RNA molecule then goes to the ribosomes, where the proteins are built. Now, once again, genes contain instructions for assembling proteins. They're containing instructions for chaining amino acids together. And amino acids chained together are proteins. And we said, once again, proteins are enzymes, which will catalyze reactions, and they also build and operate components of a living cell. Almost your entire body is made up of this bunch of proteins bonded together to make your tissues and all the cells that you need. So the sequence of bases in DNA is used as a template for messenger RNA. The codons in the mRNA special the sequence of amino acids. And let me go back to this. And then once again, you put those amino acids chains together, and then we end up with a polypeptide.